because that is just a good thing to do. Oh, did I not publish those? 1 second, let me double check. Oh, well, let me fix that. Yeah, apologies. Let's see here. That should fix it. So thank you for flagging that. That would be, don't need to make this class any more difficult than needed. Am I right? So everyone should have access to the slides, and let us start with a brief profile on a famous microbiologist, as we normally start our classes. So 1 second there. What's going on? Got it. Okay. Yes, so basically, I know we all have, had a lot on our plate, and I'm going to be sort of instituting some newer things just to help us finish strong during the last few weeks of the semester. So I hope they are helpful. And then thank you for your patience. I have not gotten a chance to put out the new survey for just checking in with all of you, but thank you for giving me a little extra time to work on that. But I do want to, just find ways to continue to support you as a class and as a community as we keep moving forward. A little bit more on that later. First off, I want to introduce you to Jorge Galan. He is an Argentinian American who earned his DVM from the National University of La Plata in Argentina, and went on to receive his PhD in microbiology from Cornell University. Later went to do postdoctoral work at Washington University in St. Louis, before becoming a faculty member at SUNY Stony Brook and then Yale in 1998. He currently serves as a professor of microbiology there and chair of the Department of Microbial Pathogenesis. In all transparency, this was a little more geared towards Micro 101 today. We're entering into our microbiota and microbial pathogenesis units, but microbial pathogenesis is also where we are at as a class as well. So I thought sort of a nice intersection there. His lab's work focuses mostly on Salmonella, and this is a figure from one of his papers looking at how Salmonella uses a needle-like apparatus called a type 3 secretion system to inject effector proteins into host cells. And so that's shown here, and then a zoomed in image of that occurring here. So sort of injecting these effector proteins into these poor host cells, and then that leads to a number of signaling events, ultimately inducing NF-kappa B activation. We've talked a lot about NF-kappa B, right? And the innate immune response that generates. And what's interesting here is Salmonella does this, and that actually induces a bunch of antimicrobial peptides that kill the microbiota, and they get sort of X'd out over here. Pretty unfortunate. Yeah, can't even see them anymore. Look at all those X's. And so basically, the microbiota are the casualties here, and that allows Salmonella to thrive while microbiota don't survive. So that is sort of the story, and one of many that his lab studies. I've actually had the opportunity to meet him. Yeah, extremely smart and passionate individual about science, and pretty much all things microbiology. And I see in the chat we have a fellow Argentinian American. That's super cool. Is it Anna or Anna? I apologize for not remembering.
It's Anna.
Anna. Okay, Anna. Great. I mentored a student back in Boston, and same spelling, but it was Anna, and it took me forever to like remember to say Anna. And now it's like now I just have to adapt to my environment, right? Just like a virus. I don't know why I'm comparing myself to a virus. Maybe it's because we're in virology. But I will try to remember, Anna, and thank you for your help there. All right, let's erase all these X's and arrows. All right, or I guess we'll just move on to the next slide. Some class announcements. There has been an updated syllabus, version 20 or version 4.26.21. And then, of course, I hope everyone noticed that all of the Unit 2 resubmissions have been graded, so we're all good there. If you would like to discuss anything, think I missed something or want to clarify some aspect, please let me know by Tuesday of next week, and we will take care of that. And yeah, just want to make sure we can finalize those things. I'm in a grading place. We'll say a grading place. And it's a busy place, and I will be grading for a while. But thank you for your patience. I'm aiming for Thursday. I don't know if I'm going to make it on the Unit 3 exams, but yeah, we will see how it goes. It may have to wait till this weekend, but I am pushing there as best I can. Our success hours will be 11 a.m. to 12 p.m. on Thursday and Friday, returning to our normal schedule there. And great shout outs on some of the posts. Your laptop makes noises when it overheats. Growl back, assert dominance. Do not let technology overthrow you. I mean, this is like so funny. Like, I don't even know what to say. I felt like it's like that could be in like Cards Against Humanity or something. I don't know. It was just like really, it was just excellent. I don't fully understand this post. It got 3 likes in response to like, do you know what tethering does? But I do know that yes, that was a big moment in the class discussion, so maybe that's why it is getting the likes. So good work there. Keep posting. I love the vibes. And also sadness, let's just go finish this degree, I guess. I don't want anyone feeling like that. I know the struggle's real. So hang in there. And wait, can someone explain mood? Like, what's like, is this just like, I still don't fully understand how to use that in like a sentence, or maybe I'm not supposed to use it in a sentence. Is it just like you say it when it's just like an intense emotion? All right, that's not like technically a full sentence. When you relate to something, oh, nice. This class is keeping me hip between used to express this something that thing is relatable. 2016. Okay, well, you know, that's great. Anyway, just a warning, when you get old, or I'll just say older, let's say older. When you get older, you don't realize it happens. Like you think it's gonna be like, oh, like I'm like getting older. Like it just happens. So yeah, I don't know. Yeah, I'll get on TikTok instead of grading your exams. Absolutely not. No way. Grading exams. Mood. Okay, I'm just gonna like say it till it actually works. Cool. So anyway, because there's this mood or vibe, right? Um, I I've oh whoops. I'm gonna like bring it up at various points in class. I thought I was gonna bring it up right now, but the energy overall feels pretty good. So we do not have to like pivot yet, but like every class is different, and I just like I'm trying to keep a pulse, my finger on the pulse of the class. So like whenever this comes up, basically we can do things. And what we'll do is an eye clicker. Oh snap! I forgot to launch. Woo. Okay, quick pause. I'm sorry about that. Have to launch. Okay, so basically how this works is classic music. Green Day is not classic rock. That's I'll stop you right there. Okay. So basically how this works is when this appears, we can we'll do a iClicker vote on the following options. So one is like music break, or sorry, that should be A. Two would be like a 1 minute meditation. Three would be like Dr. Adams trivia. And then like four would be like keep pushing, feel good, more content, please. All right. So I did this in Micro 101 and I was really shocked because like no one was ready for like material. So I'm going to do that here. And I'm totally happy to dive into the material. But if we're already like, man, this week has been rough and we need like another break or something, we can do that. But let's get a pulse of the class. And then this will like pop up at different points in class moving forward. And then we can just check in, make sure we're all in a good place and stuff. But I want to make sure that, yeah, we are able to be fresh, energized, and that we're not like just going through the motions of like listening to Dr. Adams drone on and on, but we're like actually like in it and we're like processing things and like critically thinking and engaging the material. So the poll is open. I'm going to give everyone, let's do 30 seconds and then we'll just see what happens. And there's no wrong answer here. Okay, there's no wrong answer. This is just like, can you repeat what okay, the question is, what do you need / want right now? And you pick, if you want more than one, pick your favorite, and then we can like always check back in again because the other one will like happen. But right now, where you're just picking one, and that's it. Okay. Okay. Like oh my God. You guys are ridiculous. Oh man. Okay. So this mood there, I did it, right? Mood. That's how I feel about this chat right now. On point. There. Is that an accurate use of mood? Someone tell me if that was an accurate or inaccurate use of mood. I can't give you snacks. Okay. Whoop. Dang. Okay. All right. Well, time is up. So here we go. Dr. Adams. Okay, all right. So Dr. Adams trivia is a clear winner here. So what we'll do are two trivia questions. One will be from the class, and then I will also come up with one. So do we have any trivia from the class? And then or depending on how much trivia questions are from the class, we could just do both of those. But to get us started, I guess I'll just come up with one, which is where did Dr. Adams propose to his wife? And A will be San Francisco. I'm going to keep my pattern, Jane. I saw that in the chat. It's going to be 2. It's going to be Santa Cruz. Duncan Hall. 6, 4. Absolutely not. 3 is going to be Chicama, Peru. And 4 is going to be Rio de Janeiro, Brazil. And you guys are, this is not romantic. Under the lab hood. Oh my gosh. And the last one is Mexico City, Mexico. All right. This is hilarious. This chat's the view of the west side. Oh my gosh, this is great material. I wish I could record all that. Okay. Those are the options. I'm gonna open the poll up. Let's go. Apps, you Zoom meeting. Oh, my wife does love Taco Bell, so that actually would have probably been a win. I do not, though. I would refuse to do that. Wow, this class is fire. That's accurate. I know that's accurate. Fire.
This is a clicker question, correct?
Yes.
Okay.
But I'm doing what Jane said, which is A234. All right. Last call. There's one response missing. 3, 2, 1. All right. Wow, very close. A tie between Santa Cruz and Chicama, or almost a tie between Chicama and Santa Cruz. It was Chicama, Peru, actually. So went there as part of a destination wedding for friends from Peru, actually. They're, am I gonna get this wrong? Trujillo, Peru. And yeah, we went there and yeah, proposed on the beach there where they have some of the longest left hand waves in the world. So we both love surfing and traveling. So that is where the proposal was. Oh wow, no one voted for Mexico City. Interesting. Well, I will tell you that my wife loves Mexico City. So I've never been, but I've heard amazing things. Oh, all right. Well, interesting. All right. We have time for one more trivia question from the class. What are you feeling? And then we will dive into the content. Any trivia questions? Shout them out. Or chat them out. Where was I born? Someone did not pay attention on the intro day or forgot. Is class also going to feature a guest appearance? I can do that. I think most people will get it. Oh, okay. Yeah, let's do favorite burrito. That's really tough. I'll say current favorite burrito. Or wait, I have to do answers. Duh. Okay. Fave favorite burrito. Well, my favorite food is a burrito. So fave burrito spot. We'll do that. All right. All right, Jane, back to your schematics. So we're gonna do super taqueria, then B, Cali Spartan, C, Cali wet burrito, 4 Lavix, and 5 Burrito Factory. Boom. Yeah, that's right. Can you handle that? iClicker ABC45. The poll is open. Good luck, everyone. Thank you, Jane. I'm glad I have your approval for my iClicker format. It means a lot. Whoa, Bianca throwing it down. All right. All right. Well, what does the class say? Wow, this is data. I'm taking a screenshot of this. This is the data I live by. Wait, what is? Oh, thank goodness. Okay, almost had a freak out. Forgot where Cali Wet Burrito was. Yes, it's California Wet Burrito. That there is only one right answer there. So if you said something else, see me after class. I'm serious. Like, see me after class. Like, what is this? Let's look at this data. Super Talk. I mean, Cali Spartan, La Vix. Wow. Okay. All to say is if you've gone to nachos from La Vix, absolutely not. I don't think I'm ever getting nachos from there again. I'll have to check out Andaluz. Where's Andaluz? I'll look it up later. All right. So now we know that concludes this round of Dr. Adams trivia. Hope we are, yeah, amped up, you know, just keeping it real. And I will continue to check in, make sure we're all in a good place. If we need a little break, because pushing the end to the end is rough. I hear you. Then hopefully this will help. You guys, man, well, I mean, Lavix is, yeah, but it's an institution, so you got to give it some respect. All right. Speaking of institutions, our immune system, yeah, we hope that doesn't roll downhill, or that means we're getting wrecked. So last time we left off talking about our adaptive immunity, we were talking about B cells, all right? And how B cells are able to synthesize antigens based on receptors, which are membrane bound, right? And these antigens that bind in a specific manner to these immature B cells initiate a signaling cascade, right? And that leads to the development of these plasma cells. And their lineage is shown here. And I'm not expecting you to know this. I'm just providing this as additional information. But you can see this antigen recognition event that leads to the development of activated B cells that are able to release antibodies. And we also talked about the role, the intersection between T cells and B cells, right? And this is really important, just as a quick refresher, for how our immune system, our adaptive immune system in particular here, is able to generate a robust and protective response um, throughout the duration um, of the presence of memory cells, right? And excuse me, to refresh our memory, we have CD4 cells that are really at the center of this story, because not only do they help B cells mature to plasma cells, but they also help CD8 positive CTLs, or cytotoxic T lymphocyte precursors, mature into amazing killing machines, right? And so if you are an infected cell, you're just like, put me out of my misery, right? For the greater good. And the CTLs will come in and close those eyes, right? So that is showing you how these relationships are integral to generating this robust adaptive immune response. And so I sort of spell this out in a little more detail here. Again, because we've already gone over this a little bit, I'm not going to spend too much time on it, but the key takeaways are that you have CD4 cells helping with this maturation of B cells and CD8 positive T cells. Brief nomenclature check-in here is just to make sure that that we don't confuse leukocytes versus lymphocytes. Leukocytes are referred to as a general term for white blood cells, which include lymphocytes, neutrophils, eosinophils, and macrophages, right? Whereas lymphocytes are a specific subset of leukocytes, including T cells, B cells, and NK cells. And they have this capacity to possess specific antigen detecting receptors on their cell surface, most notably the T cell receptor and the B cell receptor. And again, this is not going to be an in-depth step-by-step walkthrough of adaptive immune responses. This is more of a gentle overview, if you will, of how our adaptive immune system interfaces with the viral infection and viral antigens. And that will set us up nicely to then proceed with a primary paper on the virus of the year, and probably the decade, hopefully, where we look at SARS-CoV-2 in a primary paper. So moving forward, one important dynamic that I would like the class to appreciate is the cross-regulation that occurs when we consider what is referred to as a Th1 response versus a Th2 response. So we have these T helper cells that engage these mature dendritic cells in lymphoid tissue, okay? And these interactions will lead to the differentiation of the CD4 positive Th cells, all right? So this CD4 positive cell, it's a little immature, think like early high school maybe, and then it meets this very mature dendritic cell. It gets inspired, right? And it's like, hey, like, great, I now have some direction to go in life, right? And depending on that interaction, it will either go to college and become a MD, like a medical doctor, or it'll take the Dr. Adams route and become a PhD and spend forever in grad school and like wonder what the meaning of life is. No, they're both great options. But basically, the idea here is that there's two paths. You have a Th1 response and a Th2 response, and they're both important, and they both can help with specific immune responses. The Th1 cells are very important for controlling most viral infections. They will have a number of pro-inflammatory responses that include production of IL-2, IL-12, interferon gamma. All right, this is a canonical Th1 response. And they are often found to play the major role in viral infection, regulating viral infections. On the flip side, you have Th2, all right, and this is dominated with IL-4, IL-5, and IL-10. And here you have the antibody response dominate more, okay? And they also can influence each other, and that's shown here in this bottom chart. You have this cross-regulation by the different cytokines, right? So they will hamper or inhibit the other T cell population to enhance their own role, right? And this speaks to the complexity of cross-regulation across the adaptive immune response. So we're not going to spend too much time talking about how these balances are tilted, but I do want you to appreciate that there is this initial interaction that leads to differentiation and clear responses that are driven by a subset of cytokines that are distinct depending on if it's a Th1 or a Th2 response. So the Th2 cell response promote the antibody response by inducing maturation of immature B cells, okay? And that is important, right? Because the Th2 response in general is not really robust against fighting viruses directly. It's more through these antibody responses. Usually Th2 cells are thought to be more important for extracellular bacteria and multicellular parasites, okay? So they're both important, and more often than not, we'll be focusing on Th1s in the context of viral infections, but there are some exceptions. So when we think about this interaction, right, we have an antigen presenting cell shown here with the recognition of a viral particle, okay? It shows a virus. It's not recognizing the entire virus with the TLR. We've talked about that. But there is engagement of the toll-like receptors, and this can lead to a Th1 favored response, right? So it's favoring the killing of these intracellular pathogens. But exactly how that occurs is not clear. I also want to emphasize another sort of subset of flavors here, okay? So we have different populations, distinct populations of CD4 positive cells, right? So earlier, I tried to simplify it by just starting us off with two different flavors, right? We have Th1 and Th2, but it's actually even more complicated than that. There are many different distinct subpopulations of or T helper cells, okay? So there's 1 and 2, but there's also 17 and Treg. And each of these have been found to have distinct roles depending on the nature of the microbial infection, right? So it's sort of like if you're assembling a team of Avengers, right? Like sometimes you want Hulk, sometimes you want Iron Man, and sometimes you want Doctor Strange. I don't know, right? Like there's just, they all have different sort of abilities that they're really good at. And so these are sort of highlighted here, and dendritic cells will present antigens to help these CD4 positive cells sort of become the T helper cell that they're supposed to be. And if you look at, for example, Th17 cells, right, this will show you the distinct, or sorry, if you look at the arrows, those all help promote the generation of these specific T cell subsets. And depending again on the nature of the viral infection or the microbial invader, that's going to drive the specific subset to develop. To tie this into some of the primary literature, right? A lot of this is also really cutting edge, and there are not clear answers, right? So recently there are review articles coming out with provocative questions, right? Or maybe you don't think it's provocative, but it's trying to be provocative. The protective and pathogenic roles of IL-17 in viral infections, friend or foe? The answer may surprise you, right? So this review article is dedicated to highlighting the different pieces of evidence that talk about how IL-17 can be beneficial or problematic during a viral infection. We're not going to go through the whole paper, but I do think it's valuable to sort of lend that real world context, right? And so here are two examples from this review article. In the first example in panel A, it talks about how you have IL-17, okay, right? And what is its role in the influenza virus infection? So what we see here is influenza virus, it's attacking, all right? And where is it attacking? In our lung, right? Influenza, the flu, makes sense. And these cells, they're necrotic, they're dying. It's very sad. What are we going to do about it, right? Well, IL-17 will induce the release of CXCL13 from these dying cells. And this can then help recruit B cells, which will migrate to the lung and produce a bunch of neutralizing antibodies to stop the infection, okay? So that's one way in which IL-17 can play a role, IL-17 being released from these Th17 cells. Another example are from another subset of T cells called gamma delta T cells. They release IL-17 and they stimulate IL-33 expression, again, by these same dying necrotic cells. And this promotes tissue repair and growth of healthy cells, right? So these are two examples in which IL-17 can be beneficial during an infection. And again, the link to this paper is here if you want to check out more examples. I thought these were two fairly concise examples of how the immune response can try to help fight off viral infections. And there's, again, many more, but this gives us at least some glimpse into some of the research that has been done in this area. Another really important aspect of our immunity is the inflammasome. And one critique I have of this book is that they put the inflammasome in the adaptive immunity section. I think that this is a better fit for innate immunity, but either way, we're covering it, so that's totally fine. But I think their rationale is that there are so many activation steps here that it can like be complicated, and that leads to sort of positing it or positioning it in a more adaptive stance. But for those of you who've taken immunology, some of this will be a little bit redundant, but this is a really important mechanism by which host cells can generate inflammation and a robust immune response to infection, okay? And so this role of the inflammasome, right, is central to the production of the cytokines IL-1 beta and IL-18, which are two of the best characterized pro-inflammatory cytokines that you can get. Now, there are many different inflammasome complexes that exist. We're only going to talk about one for the interest of time. And that complex is the NLRP3 inflammasome. Excuse me. Now, the NLRP3 won't let me try to shut me down on MP3. Oh, it's MTV, MP3. It'd be so empty without me. All right, so the NLRP3 protein, which stands for the notched LRR and PYD domain containing protein 3, the adapter ASC, which is the apoptosis or apoptosis associated speck-like protein containing a card domain, right? Great short name, and procaspase 1, right? That is the NLRP3 inflammasome, right? It's a complex of proteins. This is not exactly what it looks like, but close enough for our purposes, okay? So this is a complex of several proteins that come together to help generate this immune response, okay? And there's two important signaling pathways that are required. You have maturation and release of IL-1 beta that is dependent on these two distinct signals. You have signal 1, which is initiated through the recognition of PAMPs or pathogen-associated microbial patterns. All right, so you have your TLR recognition signaling through an adapter protein such as MID88 or TRIF that induces NF-kappa B activation, right? And this leads to the generation of components for the inflammasome, including NLRP3, all right? So those are generated as well as precursor forms of IL-1 beta, see you later, don't be a hater, and pro-IL-18. So these two proteins are generated and they are in their precursor form, which means they aren't doing anything, right? And so like they still have their training wheels on. They don't do anything until signal 2 arrives, which induces assembly of NLRP3 with ASC and procaspase 1. So there's assembly, right? And then procaspase 1 is activated. So you have induction of the inflammasome, and then procaspase 1 is like, release me. And then procaspase 1 is free, and then it's like, yo, pro IL-1 beta, activate. Pro IL-1 18, activate. So it cleaves these cytokines, these precursor forms. IL-1 beta and IL-18 are released, and the pro-inflammatory reaction is happening, all right? And this all arises because of a signal 2 that can occur through potassium efflux, reactive oxygen species, lysosomal damage, right? Also, ATP can play a role here. So there's a number of different signal 2s that can happen to initiate this process. Now, my question for the class is, why have 2 signals instead of 1?
In case one fails.
In case one fails. So if one of these, what do you mean in case one fails? Like, do you mean if this TLR is blocked or something? What, can you expand on that a little bit?
Maybe.
And other students are welcome to chime in. Miriam is saying in case one is evaded. Okay. And then Timmy is saying better regulation. So these are interesting ideas. I want to draw this out a little more. So let's think about this. If, let's say that this TLR is defective, Hopefully not as defective as my Zoom skills. All right, so boom, that's gone. Now what happens? So let's say that signal 2 is happening, okay? But is there going to be any pro-IL-1 beta and pro-IL-18 around? Exactly, Timmy. There's nothing that's going to happen because we didn't get signal 1, right? We didn't get signal 1 because it was blocked. So instead you have caspase 1 that is bored. Sylvia is chiming in with multiple checkpoints based on environmental stimuli. Okay. So can you expand on that a little more? What do you mean environmental stimuli or need toxins and PAMPs? Okay, you're right. So this is a little more at, I think, the getting at the benefit of why you have these multiple signaling checkpoints, because let's say you detect, I don't know, a LPS molecule, right, from your microbiota. If that's all it takes to activate this pro-inflammatory response, then you're going to be having inflammation all the time because you're just driving this pathway, right? But we have tons of bacteria and viruses in us, right? If we're just activating these TLRs nonstop, then we're going to get constant inflammation, even though it's just our microbiota helping us process nutrients. What this 2-signal system allows us to do is really up the level of alert when there's this significant amount of damage, right? If you're having potassium efflux or lysosomal damage or a bunch of reactive oxygen species signaling, right? That means that things are really not going well, right? That toxins are inside, that you're having a bunch of holes that are like forming, plus you're getting activation. Those are all indicative of an infection, okay? And so that is really part of the value here in having multiple regulatory checkpoints. You don't want just anything to set you off. You don't want to have a quick temper. It's like someone accidentally bumps into you, you're like, ah, right? That'd be like really annoying, because it'd be hard to get anything done. This is like someone bumps into you and then like, you know, maybe they like continually bump into you or like trip you. You're like, hey, what's going on, right? So this is our immune system's way of making sure we don't freak out too quickly. All right. So I've sort of explained this a little bit already, but basically when the inflammasome links the sensing microbial products with the activation of sorry, I misspoke. The inflammasome is basically linking the sensing of microbial products with the activation of pro-inflammatory cytokines, okay? And so as I just described, this signaling cascade is responsible for generating precursor cytokines, specifically IL-1 beta and pro-IL-1 and that is downstream of the toll-like receptor. These precursor proteins are produced by signal 1, but they have to be cleaved by a functional caspase 1, which is generated through signal 2, and that leads to the formation of IL-1 beta and IL-18 as secreted and functional cytokines. Are there any questions about this process? Alrighty, seeing no questions, we're going to keep pushing. Right, and that's, again, just sort of an overview of the two different signals. And so we have a practice problem here, okay? And I want to do a check-in here. So there's, again, multiple options. I know I just gave you a bunch of content to process and navigate. And so I want to check in with the class on what feels best, okay? Because again, people aren't posting photos like this because they're like, you know what? I don't feel any burnout, right? And I want to be checking in with the class and mood. Maybe one day I'll get it right. I don't even know, but I'm trying. Okay, so we can A, do the practice problem together. B, do one Dr. Adams trivia. We can do C, a 2-minute meditation. Oh my gosh, Jane, I am so sorry. Let me fix this. Absolutely not. We can't have all letters. Not after what we've started in this class. All right, so now D will be 4 and E will be 5. All right, 2-minute stretch break or more content. Keep pushing. So think about it. I'm going to put a minute on the clock. Choose wisely. Mood. I'm just saying it. Mood. And okay, we have a direct message. Can we get a chance to do it ourselves before and go over it next class? Right. So if you want to do the practice problem solo outside of class, then I would suggest picking B, C, 4, or 5, or 5. If you do 5, that means we're going to skip the practice problem and then just do more content. Whereas A means we're going to take like 5-ish minutes right now and do the practice problem together. Another question, will we go over the practice problem at some point if we don't go over it now? Yes, I'm going to have us go over the practice problem either Thursday or Tuesday. I haven't decided yet. And if I forget, remind me, because it's not intentional. But yes, we'll go over the practice problem together as class. So any other clarifying questions before we open the polls? All right, your time starts now. All right. Get your answers in. Five more seconds. Oh wait, no, I said a minute actually. All right, 10 more seconds. 5, 4, 3, 2, 1. All right, there is a large amount of support for doing a practice problem together. So that is what we will do. All right, class. So you should have that in your slides. But we're gonna give everyone 5 minutes and then we will work through it together. And let's return to this. Okay, so this is based on some of the content we just went over. And this is, a nice formative assessment to help us all sort of see where we're at with understanding this. All right, graphing, our favorite. So you infect a macrophage with an RNA virus, you measure pro-IL-1 beta activation and potassium efflux and get the following results. Please graph the IL-1 beta levels during the experiment. And then, oh, sorry, I gotta have some erasing to do. Okay, you repeat the experiment with a different RNA virus. You only measure IL-1 beta levels and get the results below. Please graph the IL-18, pro-IL-1 beta, and caspase-1 levels. So there is a lot of different components here, and I encourage you to look over the figures we just discussed, okay, and think about how those signaling pathways intersect. I'm going to give everyone 5 minutes to get this started, and then we will check in. There are a lot of graphing components here, so we don't have to necessarily tackle everything, but just at least try to do the first part, and then maybe the IL-18 graph for the second part, okay? And I'll make sure that there is a PowerPoint version of this, so you have that as well. But I'm going to start the timer for 5 minutes, and then we will check back in after that, okay? And of course, if you have questions, please just let me know in the chat. You can direct message me or ask using the microphone. For those of you who prefer a practice problem with the PowerPoint format, that's been uploaded. All right, gonna give everyone another minute and then we'll come together as a class to go over it. Alrighty, folks. Let's go ahead and tackle this together. Do we have any ideas on how we might want to get started with this problem? What are thoughts from the class? And you can chime in on the chat or the microphone. What is the starting place or a first step?
Well, I was going to say that aisle 1B below will be the same as aisle 18 since they're like the same step, right?
Okay, sorry. Aisle one beta should be what?
Okay. So aisle 18 and aisle 1 beta, they happen at the same time in the same way. So wouldn't they be the same?
Mm-hmm. Okay, sure. So you're talking about the second part of the question?
Yes.
Okay. Right. So that's correct. And we'll spend some time talking about that in just a minute. Sorry, I think I went a slide further than I wanted to. Yep. All right. Well, let's see. Pro island beta should be more than the arbitrary levels. Wrong question. Okay. So sorry for, yeah, I'll just walk through it since I showed the answer. Didn't realize I was on the same slide. So one strategy, right, that I recommend is identifying where the different pieces of the puzzle are, okay? And so you have pro island beta data. So here you go, pro island beta, right? And you see this ramp up of pro island beta. Where is pro island beta in the system? Right here, okay? It's downstream of signal one, right? So signal one induces the synthesis of pro island beta and pro IL-18. Signal two comes in, right? That's potassium efflux. You're gonna get generation of the inflammasome as well as production of caspase 1, which will then process the pro-island beta, right? So you have this generation of the two signals, and then that should lead to the creation of IL-1 beta, okay? Again, don't focus too much on the exact numbers here. It's more about the sequencing, right? And the relationship of these graphs to one another. You should not be getting the generation of IL-1 beta at hour 1, okay, you shouldn't be getting it in hour 2. And the reason is because you don't have that second signal yet. You're not getting potassium efflux until hour 3. That means the earliest you should see IL-1 beta is hour 3. You could argue that you won't even see it till hour 4, but you definitely should not see it earlier. I have a direct message for question 1. If at hour 4 we have potassium efflux, it must mean that we have caspase. And why do we have high levels of pro-IL-1 beta? So pro-IL-1 beta does not. Okay, so let's go back here. So pro-IL-1 beta does not require any potassium efflux. You can get tons of pro-IL-1 beta just through signal 1.
I was thinking, wouldn't it start being cleaved at that point? Why would it still say would be elevated?
Oh, I see. Yeah, so that just depends on how much signaling you're getting, right? So there are many different ways to think about the generation of pro-IL-1 beta. And that's why I often will ask you to explain your rationale, right? But yes, if you think there's a weak signal induction of pro-IL-1 beta, right? Then yeah, you could have this tail off or stabilize at a certain point. Maybe you think it's a strong amount of IL-1 beta or strong signal 1 induction, in which case you might continually be ramping up pro-IL-1 beta production, even though you have now started processing some of it, right? So it's very easy to start to think about all sorts of possibilities. And as long as it's consistent with the signaling pathways in this model, right, then we're able to generate logical graphs, and I'm able to give you credit for it. So if I was asking you what the pro-IL-1 beta levels look like, you could have it increase, you could have it stabilize. Debatably, you could even have it decrease after a certain point, right? So there are multiple options there. If I'm asking you about end results, though, then you definitely want a situation where you have IL-1 beta that's not produced until the earliest, I'd say, is hour 3. Checking the chat for any other questions. I don't see any. Coming back to what Suzanne said and Timmy, I believe, for the second question, right? So we're asking about IL-18. Pro-IL-1 beta and pro-IL-18, they're friends. They like to hang out together. They go on walks together. They go bowling together. Like, it's just a blast, right? So when IL-1 beta gets cleaved, or sorry, when pro-IL-1 beta gets cleaved, pro-IL-18 is getting cleaved. Caspase 1 comes through is like, hey, you guys are ready to go out there and save the world. And they're like, oh, we were just going to go bowling again. And Caspase 1's like, no, actually, you're going to go save the world. They're like, all right. They get cleaved, they get secreted, and then they're off to help generate that pro-inflammatory response. Okay. Likewise, your graph should have a similar trend. You could like make the case that they don't have to be the exact same arbitrary levels, but the trend should definitely be the same. Okay. For pro-IL-1 beta, all right, this, again, thinking about the timeline and the sequence, you want pro-IL-1 beta to be present before IL-1 beta, okay? So if we have a system where you're having an increase, a consistent increase in pro-IL-1 beta, that's fine. This could vary, but you definitely want to have pro-IL-1 beta generated prior to IL-1 beta, all right? Because that's what happens. That's how that timeline works. You can't get IL-1 beta until you have pro-IL-1 beta. Lastly, caspase 1, same difference, right? You're not getting IL-1 beta until you have caspase 1. You're not getting caspase 1 until you have pro-caspase 1, okay? And so again, thinking about how these pathways are intertwined and interconnected is really important for how this pathway functions. Question in the chat, can we show pry-IL-1 beta decreasing as IL-1 beta increases? Yes, you definitely can, right? Some of this is just going to be how you envision the system. If the signaling pathway induction process is transient, there was a signal, the signal went away, like you have a limited or finite pool of pro-IL-1 beta, then that can decrease and I can give you credit for it. But I can't give you credit for having caspase 1 after IL-1 beta, right? Like you can't have caspase 1 and show up at hour 3 if you're having IL-1 beta show up at hour 2. All right. So that brings us to a close for today. I'll stick around if anyone has a quick question or two. Thank you for your participation and look out for a check-in, optional check-in survey. And I hope you have a great rest of your week.